Okay, it looks like it's going to get started soon. Looks like it's working now. Great. So good morning, everyone in the United States, afternoon, evening to those of you in Europe and other parts of the world. It's nice to see folks here. I see a lot of familiar faces, and thanks for joining us for today's FIND seminar. My name is Lauren Hayes. I'm one of the co-hosts and the coordinators. Okay, it looks like it's going to get started soon. I did it again. Looks like it's working now. Great. So good morning, everyone in the United States. I did this last week, so I'm just trying to replicate the situation. Get up my sample size. I'm messing up the live stream to YouTube. Anyhow, again, my name is Lauren Hayes. I'm at the University of Tennessee at Chattanooga and hosting the seminar today. I have a couple of announcements that I always do before introducing the speaker. First, I want to thank last week's speaker, Lena Grinstead, for a really cool talk on social spiders. The talk was fascinating, and we actually had a lot of discussion that occurred during the formal discussion. And of course, afterwards, we stuck around for another hour talking. So it was really a really nice seminar interaction. Next week, we have the last speaker of this this particular year, the spring, I'm sorry, and that's going to be Becky Rosengass, who's at the Northeastern University, and I believe she'll be talking about her research on eusocial insects. We have a seminar series, and Nancy, could you just stop sharing your screen just for a moment? We do have a series coming up. We've really made a lot of progress on, I think, the schedule for the upcoming fall. Can everyone see this okay? Can someone just give me a thumbs up if it looks good? You should see the schedule should be coming up. And so you can see here, we haven't posted this on our website yet, but we've got a great list of people coming in giving talks. And you can see it ranges from folks talking about, Dr. Gadigar is going to be talking about paper wasps. We got Shengfeng Shen from Taiwan talking about burrowing beetles. Jen Smith from Mills College studies carnivores. Juan Carlos Ribareta studies birds. Clint Pennick from Kennesaw State, which is just down the road from me, is going to talk about dominance hierarchies and brains in ants. Annalise Beery is going to give a more mechanistic talk on the neurobiology of social behavior. Then we have Andy C. talking about personality. Julia Fisher from German Primate Center will be talking about social evolution in baboons. Monica Bergerhoff-Mulder will be talking about human behavior. We then have Wolfgang Goeman talking about birds, mating systems, and physiology, followed by Hannah Coco, social evolution and life history theory. Noah Pinter-Wollmer from UCLA will talk about spatial and ecological effects on collective behavior, and we'll wrap it up in late December with Valeria Romano from Strasbourg talking about social networks in the Stone Age. So a pretty cool set of seminars. We have a couple that we're still working out, and when that seminar, when we finalize that, we will we'll have that posted on the website and send an announcement out to everyone so that you can plan to attend. Okay. So Nancy, if you want to share your screen again, I'll introduce you, and really excited for today's speaker today. As you all know, many of you know Nancy Solomon from Miami University. Dr. Solomon was my PhD advisor, and we maintained contact for all these years. I think it's been almost 17 years now. So Nancy's earned her undergraduate at the University of Pittsburgh. She then did a master's degree at Case Western Reserve University in Ohio, and then got her PhD at University of Illinois in Champaign-Urbana. After doing a postdoc at North Carolina State with Vandenberg, I believe, she then got a job at Miami University, where she's been since then, where she went up the ranks from assistant to associate to full professor. Nancy is a really well-known person in the field. even when I was, looking for grad advisors. I knew Nancy from her work with Prairie Voles, which you'll hear some today. Her research is, takes an integrative approach to understanding animal behavior. Her work focuses on mating patterns, cooperative breeding, sociality, and she looks for relationships between fitness and mechanisms using both field and lab approaches. And as you'll see, she has this really unique site at Miami University where they can actually do experimental manipulations in the field, which is really nice. And for most of her work focuses on these prairie voles, but she's also done some work on feral cats and other species she may talk to you about either in discussion or in the talk, not sure. She's funded her work consistently through the years, both through the National Institute of Health and the National Science Foundation. And she's been really productive. She's published about 100 publications in really good journals, all the major behavioral journals, molecular ecology, many in good journals like Journal of Mammalogy and Tree. And I think one of the things that really drew her attention to me was her book on cooperative breeding in mammals. That's a really well-regarded book from, I think, the late 1990s that I still use in my readings. So Nancy also is a leader in the field. She was an editor at the Journal of Mammalogy and Animal Behavior, and now she's one of the 2 executive editors at the Animal Behavior Journal. And I can say, one sort of a personal comment, Nancy was a phenomenal advisor. She really gives her students a lot of time and energy, and she's attentive. And what's been really fun is after being a great advisor, she's evolved into a great colleague too. We've done some work together through the Fine and also on other projects. It's been fun working with her on those. And today she's going to be talking about her work on prairie voles. And the title of her talk is Interpopulational Variability in the Prairie Vole Social System. And with that, I'll turn it over to Nancy. And thank you again for joining us today.
Thank you, Lauren. That was a pretty amazing introduction. And now I'm going to have to live up to some of those words. And thank you all for joining me as I get to talk about one of my favorite topics, social evolution and variability in the prairie vole social system. So I worked on prairie voles for my dissertation, took a break, worked on another species of vole, but found that they were much harder to work with here in Ohio and outside of the range pretty much of the pine voles, since I couldn't find any here. So we went back to working on prairie voles because we're able to work with them in the lab, in semi-natural enclosed populations, as I'll show you, and in some natural populations. So we could look at behavior in the lab, and then we could get a taste of what goes on in their real world. So today is divide this talk into 3 parts. First, give you a general introduction to the topic, brief one, and then talk a little bit about the star of the show here, the prairie vole, Microtus ochrogaster. Then talk about a couple studies we've done looking at intraspecific variation between populations, and then end by talking about intraspecific variation within populations, but also using that to discuss the effects of some of this variability on reproductive success of prairie voles. So prior to the 1960s, a lot of species were characterized as having social systems that were species specific, or in other words, a particular species had one type of social system, and any variability that was found from that was considered to be noise in the system or some kind of pathological problem, or neutral from the perspective of selection. So it was kind of ignored. But we know more now. Today we talk about social systems as being found in particular populations under particular circumstances. And I'll try to use the language that Kapler talked about in his 2019 paper, where he broke down the social system into 4 components, including the social organization, which included things like group size. So we could say that a population consisted of a lot of solitary individuals, maybe some male-female pairs or groups. And I define groups as social units that contain more than 2 adults. Another component of the social system is social structure, which he defined as the social interactions among conspecifics. And then the mating system, or as some of the literature calls it, the genetic or reproductive mating system. I'll just use the term mating system today. So who mates, or in a lot of cases, we can only discern who reproduces using parentage analyses. So again, things like monogamy, or multiple mating by both males and females, then polygynandry or promiscuity. And what I don't have time to talk about at all today, but there's some interesting things going on with the prairie voles, is the care system. So who cares for the offspring that are born to one or more females, depending on the species or the population? So, as I said, today, I think most people would say that a particular population, for example, is monogamous under these conditions or is not. Or, as I'll show you later in some of my slides, break down the types of social units and or mating system in which the type of mating system we see in a particular population. So a certain percentage of individuals display different social organizations, for example. And we know that in general, often the first response to any type of environmental change or challenge is a change in behavior, which then subsequently affects other aspects like the social structure seen in different populations. So in habitats that have changing or unpredictable environments, variability or flexibility in the responses or behaviors is often favored by selection. And this could either be temporal variability or geographical differences, so changes over space, such as finding differences in different populations. So today I'd like to take a peek at some variation between and within populations, and I'd like to introduce this using a diagram from Karsten from his 2013 paper. So if you first look at panels A over here in the top left and B in the top right, you can see differences between populations that in A could be due to genetic differences between individuals in population 1 and population 2, or due to differences in some kind of environmental factor that leads to differences in the display of social organization in population 1 or 2. If we look within a population, as diagrammed in D here, we see that some kind of stimulus or variable affects the behavior of individuals such that we see one type of social organization or mating system at one point in time. And then if there's a change in the environmental factor, due to phenotypic plasticity, we see a different type of social organization. So today I'm not going to be able to talk at all about variation among individuals. I'm going to treat them more or less as homogeneous for the purpose of this talk. So if you look at the literature, over decades, intraspecific variation has been described as having a number of different potential correlates or determinants. For example, some ecological factors that might affect social organization or mating systems could be critical resources like food. So investigators have looked at distribution of critical food resources, food quality, or the abundance of particular food items that are critical to the diet of those individuals. Habitat characteristics is another potential correlate. For example, we may see different social organizations in an open versus a sheltered habitat. And then predation can differ on a geographic scale for sure, and so can have an effect on social organization or mating systems. Another factor could be a demographic factor, for example, population density or adult density in a population, which could result in filling of the high quality habitat, often referred to as habitat saturation. And that would lead to competition for resources among individuals in the population, again, which could affect the social system or mating system of those species, of the individuals in that population, sorry. So as an example, let's look at some stereotypical prairie voles here and two populations that differ in resource distribution. So in the one on the left, I'm sure you could see if you look really, really closely that the herbaceous plants are uniformly distributed in this population, whereas in the one on the right, you see that they're clumped in their distribution. And we could see differences in social organization between the two. So on the right, the uniform distribution could lead to pairs, male-female pairs, inhabiting that population primarily, whereas in the one on the right, we would see the formation of groups because each patch or clump could potentially support more than two individuals. So in the literature, there's been quite a number of models proposed that predict this kind of pattern, either an increase in group size, where resources are more clumped, or a change from monogamy to polygamy. And one good reference where these sources are reviewed is a paper by Chris Mayer and Robbie Berger, published in 2011, that talks about some of these specifically. So the prairie vole, Microtus ochrogaster, is found in north central North America. It's thought to have evolved in the plain states, which are about here, and then the range has moved east and west to the Rockies with the expansion of the grasslands. So the habitat that prairie voles evolved in was filled with prairie grasses or monocots with some scattered herbaceous plants referred to as dicots or forbs. And lab studies conducted by George Batsley and students have shown that these herbaceous plants or dicots are critical in the diet of the prairie voles and often patchily distributed within a habitat. We know that prairie voles also need cover as protection from predators. And I can't remember who said this, I should, but I can't, because prairie voles are considered to be essentially the potato chip of the prairie. So a predator can't just eat one. And they have lots of predators, both terrestrial and avian. I'll tell you about a few in a minute. So prairie voles currently occupy a variety of grasslands in north central North America, in that map you just saw. And some of the predators include small to medium-sized terrestrial mammals like weasels, foxes, and bobcats. Also birds of prey or raptors like owls and hawks, and snakes. And when I was a graduate student in East Central Illinois, we'd be out in the field and we tended to see things like black rat snakes fairly commonly. And the issue with those to a prairie vole is that even though the prairie voles have sometimes made underground nests, the snakes can get down into them. So another thing about the habitat that the prairie vole occupies is that there's some unpredictable features to it. So droughts are relatively common. It's thought that they occur about every 2 to 5 years, like a semi-major or major drought, but the onset is not predictable very far ahead of time. And then there's drastic changes in the population density of prairie voles, as you could see here from this graph showing the dates or years on the x-axis and population density on the y-axis. So populations could go extinct or be very, very small, close to zero when they're at low density, or increase until they're into the hundreds. And the one year when I was there, we had a banner year for prairie voles where we had over 600 individuals per hectare in a population. Needless to say that we couldn't finish one trap check before the time for a second one 3 to 4 hours later began, with that many animals going into our traps. So Lowell Getz, who spent the majority of career at the University of Illinois in scenic Champaign-Urbana, is shown here in the field with some of the traps he used early in the study. And he's well known because he's the first one to have speculated and then described the social organization and mating system of the prairie voles. And the quote at the top is from a sonnet that somebody wrote about this research. But what he found in the populations he studied in East Central Illinois in various habitats, that the herbaceous plants or dicots were prevalent and fairly uniformly distributed in this habitat. Surprisingly, he recaptured the same male and female together in a trap, in multiple capture traps, multiple times, which was totally unexpected, because when he was also studying meadow voles in these same habitats, he never caught more than one individual in a trap at the time, never male-female pairs together multiple times. These males and females that were caught together also displayed a high degree of home range overlap. So their home ranges almost totally overlapped each other, but not the home ranges of other conspecifics. And his field data showed that these individuals typically survived in the field for a year or less, again, because of predation. In the lab, they could live for 2 to 3 years once they're protected from predation. He had a collaborator, long-term collaborator there in the psychology department at the University of Illinois, and a number of you might have heard of Sue Carter. She and her students conducted lab studies that were based on some of the findings of Lowell's from the field. And the one thing that they showed, which was really important for understanding social organization, is that males and females that were together for a significant period of time and or had mated showed a significant preference for their familiar partner. And you could see this in this figure in the upper right-hand corner. So if a male and female were housed together for 6 hours, there was no mating, the individual did not show a preference between spending time with its familiar partner versus an unfamiliar conspecific. But if they were together for 12 or 24 hours and or mating had occurred, then both males and females would show a significant preference for their familiar partner as opposed to the conspecifics. And this was in a simultaneous choice test in this apparatus shown at the bottom of the diagram. So Sue was able to, one of the things she and her students added to our knowledge of prairie voles is that these voles appear to form attachments or bonds with their partners. Today, the prairie vole has become much more popular, studied in a number of labs, and currently most of these are examining prairie voles as model systems to study the neuroendocrine core of bond or paternal behavior and things like that. So in terms of intraspecific variation between populations, which is what I'd like to start off with, again, I'm showing you the distribution of prairie voles from a classic map that's published in the Biology of Microtus book. The pink star shows where Lowell Getz worked and the populations he studied. We established 2 additional sites, one in northeastern Kansas, just outside of the University of Kansas in Lawrence, Kansas, and one in south central Indiana near Bloomington, Indiana. And we selected these sites because we expected to see differences in the habitat there. And we knew that there were populations in these areas due to previous demographic studies by other vole biologists. So as I said, vegetation or population density can affect social organization or mating systems. And this could be partly because of the way males and females use space. So again, if you look from left to right here, if vegetation or some critical component of vegetation like dicots is widely dispersed, we would expect to see home ranges dispersed and non-overlapping, and the voles might either be solitary or pairs. But if dicots are clumped, we would expect to see either pairs or groups, because these clumps, again, could support multiple individuals. So these patterns of space use, then, we expected to result in differences in social organization and or mating system between populations. And again, the 2 sites we selected for this study that I'm going to tell you about right now was Lawrence, Kansas and Bloomington, Indiana. We conducted vegetation sampling initially at these sites. And in Kansas, the vegetation, the dicots were not clumped and neither were nest sites. But in Indiana, we found that dicots were clumped and so were the nest sites of the prairie voles. So for all of these studies, I'm going to give you very, very little bits of methods for those people that don't work with rodents that you can't see in the field. And then I can answer questions later if you have some. So we studied each population for 2 years. We had a little bit of overlap, but not total overlap due to logistic circumstances. So Kansas in 2005 and 6, the Indiana population in 2006 and 7. We studied prairie voles by using multiple capture live trapping. And we set traps some weeks on a grid, like a 10 by 10 grid within our study area. And other weeks we trapped right at the nests, entrances to nests or runways around nests using these multiple capture traps. And this allowed us to capture offspring and get a better idea of who was living with whom. So the next few slides are set up much like this, with the two populations on the x-axis, Kansas and Indiana, and then the variable, the dependent variable, in this case, it's home range size, on the y-axis. For most of these slides, males are shown with the gray bars and females with the white. So in our analysis, we found that the best predictors for home range size was either site and sex, or an interaction between site and sex that you could see here. So in Kansas, there was no significant difference between the home range sizes of males and females. But yet in Indiana, males had home ranges about twice as large as those of females, allowing them to overlap, potentially overlap more than one female. And this could affect both social organization and mating systems. So this slide is a little bit different, but Indiana, instead of being gray, is black, and Kansas is white. The x-axis in this particular slide shows the type of social units in which we found females living in these two populations. And site and population density were the best predictors of the types of social units in which we found females. And you'll notice that in Kansas, most of the females were single females, some pairs and groups. But in Indiana, we saw the opposite, where the majority of females were living in groups with more than 2 adults, and then the smaller percentages lived as pairs and fewer as single females. In terms of the mating system, this slide shows the proportion that were monogamous in Kansas and Indiana. Again, the gray is the males, white is the females. So population density was the best predictor because population density in Kansas was low to moderate and Indiana was moderate to high. And this is slightly confounded with site, as I'll mention again later, because we only had high density in Indiana and only low density in Kansas, but we saw moderate densities in both populations in 1 year. So in Indiana, we didn't find male prairie voles defending groups of females, but instead found polygynandry. So there was multiple mating by both males and females, as opposed to the high percentage that were monogamous in Kansas. So to sum up this first study, in the Kansas population, we found that more females were solitary and monogamous than in the Indiana population. And remember that in Kansas, the vegetation was more uniformly distributed. I didn't tell you, but it also wasn't as high. There were fewer dicots in general, and the density of voles was low to moderate as compared to moderate to high in Indiana. And I mentioned the confound between population density and site. So one of the things we weren't able to detect in this study would be to see if social organization and mating system responded to changes in an environmental factor. And so to do that, we conducted a common garden experiment. So we trapped adults for this study in either Kansas, that same population I just told you about, or Illinois, instead of Indiana, brought them back to Oxford, Ohio, bred them in the lab till we had F2 individuals that we could release into our field enclosures shown here in the lower left. So by breeding them in the lab, we were able to hold their postnatal environment somewhat constant. Differences could be in parental care. We also, by conducting this common garden experiment, were able to keep population density the same in the enclosures in which we released voles descended from Kansas or in Illinois. So these enclosures are 1/10 of a hectare. If we release 80 per hectare, which is 4 males and 4 females, there's more than enough space for their typical territories or home ranges in these enclosures. And then sex ratios were equal in these populations. Vegetation was similar since we planted the site number of years previous with a vegetation mixture based on what prairie voles used and needed in the field. We were able to have 7 replicate populations from each site, and we studied them in the study for 8 weeks. So I could sum up this slide and the next slide basically by saying we did not find differences in a number of measures of space use, mating success, or reproductive success in voles descended from either population. So if we looked at male space use during a period early in the study or later in the study, we found no difference in the home range size of males descended from Illinois versus Kansas populations. No difference in the distance that males moved. No difference in the proportion of females in that enclosure that each male overlapped home ranges with. And no difference in the proportion of nest sites in that enclosure where males were trapped. Similarly, on this slide, I have the means for you, most of the means and the p-values. And you'll see again that none of these are significantly different. So if we look at indices of mating success, the number of females that males transferred ultraviolet reflective powder to was not significantly different. And basically what we did is we powdered males, as shown here, with ultraviolet reflective powder. If they encountered a female and interacted with the female, the powder would transfer from the male to the female. And we were able to see that they interacted, and depending on the location of the powder on the female, to get an idea if they had attempted to mate with her. We also could see if they entered nests because we would find a particular color of powder corresponding to a particular male around the entrance to a nest, and there was no statistically significant difference there. In terms of reproductive success, the probability was 5%. So males in Illinois sired slightly more offspring. Sired offspring with slightly more females than did males descended from the Kansas voles. But again, the p-value was not less than 0.05. It was borderline. Make of that what you will, but you could see that if we looked at the number of litters or the number of offspring overall, there was no significant difference in males from these two populations. So when housed in the same environment with the same vegetation at the same population density, we found no differences in space use, which suggested, before we even analyzed the rest of the data, that there was no difference in the mating system between males descended from the Illinois or Kansas populations. We also found no difference in the indicators of mating success, the number of individuals they mated with, or in the reproductive success, that is the number of offspring that we were able to capture among males from the Illinois or Kansas populations. In another study that's similar but somewhat different, using different populations in part, Ophir and colleagues found no differences in their measures of social organization and mating patterns in voles from a population in Tennessee as compared to the same population, male animals descended from the same population in Illinois, in either lab studies or field enclosures similar to ours. So the results of these studies, both of them suggest that the differences that we saw in the populations, Illinois and Kansas here, but also the Indiana population, could be due to an environmental factor, suggesting that the social structure is flexible. Okay, for the last part of this talk, I'll tell you about two more studies where we look within populations, again, at some of these possible determinants, and we're going to look at some slightly different ones here. But in terms of ecological circumstances, we know that territory quality can affect group size. So we're going to look at group size as a potential correlate of intraspecific variation and how it affects reproductive success. We know that things like population density could affect survival, so that will be another factor we'll look at. And then in the last study that I'll tell you about briefly, social factors like interactions with conspecifics can affect relationships among these individuals and the formation of bonds. So let's start off by looking at a study that we conducted in the same Indiana population, again, shown here. This was in 2010. So same site that we used before. In this year, we were able to study the animals for 15 weeks using methods similar to what I described before. And then we used parentage analysis to look at the mating system, and we were able to assign almost 60% of the offspring to both parents. And that's about what people can usually do in natural populations, because some of the adults we capture might live off the study grid or disperse at some point. So to start, this is similar to what I showed you before, but slightly different. This is the types of social units for the females that we found in that year, 2010. And the first three here on the x-axis are the types of social units you saw in a previous slide. So solitary or single females, male-female pairs, or groups with more than 2 adults. And this is the same pattern, that the majority of females were found in groups as opposed to pairs or living solitarily. But I've also shown here that about 25, less than 30% of females changed between different types of social units during the 15 weeks of the study, showing that the same individuals can be flexible in terms of the social units that they inhabit. So in these populations, we see what we define as residents at a nest. And those are individuals that are found frequently at only one nest, usually more than 75% of the time at that nest. And then sometimes they have to be caught every week as well, to be considered a resident during that time period. So about half of the females were residents during this entire 15 weeks, not necessarily always at the same social unit, as I showed you. The other females, we would either not be able to assign or were considered to be wanderers if we caught them frequently at, let's say, 2 nests. So this figure here shows the frequency of social units for females that were resident the entire time. And the point I'd like to make by this is that they tended to live as pairs or in small groups. For the most part, the largest group we found consisted of 6, a combination of males and females, but we found very few females in these very large groups. So we also found the group size did not predict female reproductive success. Again, if we plot group size and number of offspring that we're able to assign parentage to, and we see a slight negative tendency, although this is not statistically significant. Group size also did not predict female survival. But the time that the female spent as a resident of some type of social unit did predict her female reproductive success. So females that were residents for the entire study, which was composed of 4 nest trapping periods, had produced significantly more offspring than those, for example, that were only caught at 1 nest trapping period. And some of this could have been that they were only on our grid for a certain period of time and we didn't catch some of the offspring. But we also found that survival, again, which could be slightly confounded with time spent on the grid, predicted female reproductive success. And this was a positive relationship. So time as resident and survival for females that were resident the entire time or even part of the time predicted female reproductive success. So instead of group size, but we weren't totally surprised by this because there were other studies suggested for prairie voles that living in a large group did not increase reproductive success. Like I said, we found a negative relationship between group size and female reproductive success, even though it wasn't statistically significant. And McGuire et al. In the population in Illinois found this same pattern. And then we found this in other natural populations as well. Group size also didn't affect female survival, but time as a resident and survival did predict the female's reproductive success. And it may be that maybe if the female's a resident at a nest, instead of moving between 2, for example, she might have more time to nurse pups or have better access to a food cache, which is also found in these underground nests, than females that were wanderers. And those that survived longer had more time to produce litters. So in the future, what we want to look at is what factors are there, particular environmental or demographic factors that affect female survival and residency. It's likely that density could have some effect on residency, for example, but we don't know the direction of the relationship yet. So the last study I'd like to talk about, again, took place in our semi-natural enclosures, and in these, in collaboration with some colleagues from the University of Michigan, in particular a graduate student, we were able to look at some interactions in the field indirectly. I was telling Lauren earlier today that, I'm jealous of people that could actually see their animals, like Dan Blumstein, who published a useful paper in 2013, and then Lauren's got one with Tina Way, with the Dagoos, where they were able to actually see the type of interactions that go on, in particular in the yellow-bellied marmots. But all we're able to do with the prairie voles, which are either underground or under the vegetation, is estimate interactions by using these passive integrated transponders, which you could see here, graduate students injecting one under the skin and the scruff of a prairie vole. And then we have monitors, handheld readers, as well as stationary readers, somewhat stationary readers, that we could use to determine where an individual is, when it's there, and what other individuals are nearby. Sorry. Always helps to go in the right direction. So again, these are enclosures at Miami's Ecology Research Center. This preliminary study, the student monitored 2 enclosures, one at high density, one at low density within one of the sets of enclosures. And this was limited by the number of readers that we could purchase. So we purchased these RFIDs, mounted them, and positioned them in one of 2 arrays within each enclosure. So we changed these every few days. And then basically what she did was conduct social network analysis to look at the number of interactions. So we did find differences in survival of voles, such that those that were in the very high density enclosures had survival that significantly decreased over time. And we only saw a small number of young produced. The gray markers and lines indicate the males and females in the low density enclosures. And you could see that they survived pretty well and produced lots of offspring. But of the founders, there were no differences in the sex ratio over time. So these figures are based on the data from the social network analysis. And for those that don't use it, when we talk about interactions, I'm using the word connections because we don't really know what they're doing. We know they're close to each other. And the term used in social network analysis is degree. So when we say degree, we mean a connection. So these figures show the number of males and the number of connections on the left with all voles, same and opposite sex. Females are red, males are dark blue. And then on the right, the number of connections with only opposite sex voles. So again, females in red, males in blue. And you could see for both of these, there's curvilinear relationships, and the best predictors were sex by degree squared explaining the shape of the relationship. And this is for both all individuals and opposite sex individuals if we look at mating success or the number of mates for each of these individuals. If we look at reproductive success, we see a very similar pattern. And again, this is all voles, opposite sex conspecifics for females in red, males in blue. And for all individuals, degree squared again was significant. There was no sex difference. And sex interacting with degree was almost statistically significant when you look at opposite sex conspecifics. Furthermore, the voles in the lower density enclosures had greater reproductive success than those in higher density, as I showed you in that first figure in this part of the talk. The other thing that Sable looked at was an index of condition of the voles, and she used body mass. So the average mass, since voles were caught repeatedly, again, compared that to the number of connections with all voles and same-sex and opposite sex conspecific. So she just looked at males here, and found that degree square, the number of connections squared, was statistically significant for all voles and females only. And body mass was not affected by density or survival. So the pattern was, again, that the voles with either most or the fewest connections weighed the least. Those with an intermediate number of connections weighed the most. So to summarize this last little bit, voles in the high density populations had lower reproductive success, possibly due to lower survival. The variable survival was linked to mating success and reproductive success. And so we also saw that an intermediate number of social connections with all individuals was the best predictor of both mating success and reproductive success. And we were only far number of connections, do an analysis like Lauren and his colleagues have done on the strength of the connections. So we don't know about that, but it could be that some balance between the number of connections and the strength of the connections may have resulted in the best mating success. And I'll talk about that a little more. So we also found that the males with the most connections might have been in worse condition as indexed by body mass. And this may have been due to the cost of territory defense or conflict by living in a larger group. So this might reflect a trade-off between the number of social interactions a male engaged in and the time that they had for foraging or paternal care, since males spend about an equivalent amount of time caring for young as do the females. And as I said, we're only looking at the number, not the strength. So we don't know if the interactions were affiliative, neutral, or agonistic. So some conclusions from this talk. I hope I have convinced you that prairie voles do show interpopulation variation in social organization and mating system. This phenotypic plasticity, we think, may be either directly affected by the distribution of something like dicots, these herbaceous plants in the population, or by density. Or it could be an indirect effect. For example, the distribution of dicots could mediate the differences we see in the mating system in the different population through their effects on population density. And we're able to manipulate density in the enclosures. So we could look at that some more. I don't know if we'd want to attempt to do that in natural populations. So the intraspecific variation in reproductive success that we saw seemed to be best predicted by female survival and the number of connections males and females had with conspecifics. And we're proposing that it's important to have a balance, that these individuals need a balance between the number and strength of connections. So for example, individuals with an intermediate number of connections might have more affiliative interactions that are strong enough to result in mating. And so that might be why they have the highest reproductive success. And Joan Silk and colleagues have shown that in other ways, strong affiliative interactions are important in baboons that she studied and may also be in some other primate species. So on the other hand, if there's too many connections or interactions, then the strength of all of them might decrease. There might be fewer matings and thus lower reproductive success. So in the future, one of our plans is to identify some of the traits that affect survival and reproductive success, because this will also provide us with more insight in understanding adaptation to the grassland habitats by species like the prairie vole. Right now, we've talked mainly about functional significance, but as suggested by a number of people, well, not initially Ernst Mayr, but also Tinbergen's got the famous paper in ethology, that we also want to look more than we have, and I haven't been able to talk about that at all, at genetic factors, developmental influences, and evolutionary influences on intraspecific variation, because if we're able to ever fully examine these, we would be able to move on from this functional approach that we've taken so far and obtain greater insight into variation of social structure and subsequent reproductive success, at least in the prairie vole, and see then through some comparisons how this corresponds to what's going on in some other species of rodents as well. I think you realize that although I'm good, as Lauren said, I'm not that good and couldn't have done that all myself. So I've got other faculty members at Miami, University of Michigan that have collaborated, former postdocs, graduate students, in numerous, too many to mention, undergraduate vole wranglers that were out in the field day and night, drought and rain, as well as excellent advice and help from field managers at Miami University, as well as Indiana and Kansas. And then, colleagues at the Center for Bioinformatics that were a great help, particularly when we were starting out with parentage analyses. So at this point, I hope I didn't run too long, and I'd like to thank you for your attention, and I'm happy to answer any questions now that my short tale is done.
Thank you, Nancy, for that great summary. Fun to hear about what's going on with the prairie vole work. If you could please stop sharing your screen. And what we can do now is we'll have time for questions. And as always, the way the question and answer session works is if you have a question, please put a question mark into the chat. And I will call on you. If you're on YouTube, there will be a bit of delay. I'm watching YouTube. It's a couple minutes behind, but if you have a question there, please post them there and I will relay it to Nancy. So if you have questions, throw a question mark into the chat and we'll get started here. And I see we have one from Kaya Tombach.
Hi, thank you so much for the great talk. I'm Kaya Tombach. I'm a postdoc at Hunter College in CUNY in Jessica Rothman's lab. I noticed that, so it looks like most voles, when they go from very few connections to an intermediate connections, get a boost in reproductive success, but some definitely get more of a boost than others. And I was wondering if in any of your other work, did you see evidence for social stratification? Was there, an emergence of dominance, differentiation as soon as you get beyond, a group of 2 or 3?
What an excellent question. Unfortunately, I have no idea because we haven't looked at that either in the lab or field. I had a previous graduate student looking at some dominance interactions in a totally different species, but I can't extrapolate from one to the other. Sorry.
No problem. Thanks.
Here, our next question comes from Ricky.
hello.
I get to see you twice this week.
I know. It's very exciting. I love hearing. I also work with voles over at Cornell with Fear, who's mentioned. I always get excited. We're still working with voles. So my question to you was about the groups that you talk about a lot. Do you know how interrelated they are? So rather, how many of them are kin?
What we think, we partially know this, but we haven't fully answered this yet. During the main breeding season, which is spring to late fall, we think the groups develop from a male-female pair and litters of offspring. So there could be individuals that are sexually mature there from a previous litter that remain philopatric. They stay at the nest and help then rear their younger siblings, as in huddle over them or lick them and groom them or something like that. So they're pretty closely related. Voles also form groups in the winter, at least in East Central Illinois, where Logets would be out year round. And he and his students would go out in their snowmobile suits in Illinois. You know what that's like, Ricky. And nobody's done the relatedness analysis on winter groups. We know they're larger. Based on his trapping data, we think that they're probably less related, but that's something that now the voles we have in the enclosure stay there year round. We could do that, but that wouldn't really be the ones to use. We really need natural population to look at this. So for your postdoc, if you want to come here and travel to the Midwest, it's very scenic.
Possibly. I am from here. From there. It is quite scenic.
I'm not pushing or anything. You could do this as a side project. But that's still something we'd like to know. We expect that the winter groups are less related, and sometimes they breed in the winter, sometimes they don't. So
I know we see that in meadow voles, which aren't nearly as social as the prairies, that they do still come together over the winter. Do you know by any chance if those guys are kin, the ones that the meadows?
I don't know that anybody's looked at that either. I know Annalise Beery studies a lot of the mechanisms in the meadow voles, but I don't think she's done field work with them. So that actually we could do in the field here because we've got meadow voles everywhere in a good year.
Thank you.
All right, so the next question in the queue is Marin. Marin Hook.
Maybe we could come back to
Yeah, I'll come back to you, Marin. I'll check back in after the next question. So I had Luis sent a request for a question to me. So go ahead, Luis. Luis, you're on mute.
That's the best kind of questions, but hard to answer.
Okay. No, thank you for the very nice and interesting talk, Nancy. It's nice to see the whole picture, or at what time it's now going on. So my question is, I got obviously puzzled by the one of the findings in which you have this. During some years you have this number of these voles that they form bigger groups. My understanding is that those groups include multiple females and potentially multiple males, of course, as well. And so you didn't find any evidence that thus far that the formation of these larger associations or social groups attain obvious fitness benefits. That's what I get, what I got. So my question is, since you also at the very beginning of your talk, you mentioned how ecology or ecological conditions overall in your sites were also very variable when you look at inter-year variation. And so you have droughts there that seems to be recurrent every 4 or 5 years, if I recall correctly. So I wonder, question is then, have you start looking at how this ecological variation may help explain a little bit some of that puzzling situation? I mean that, for instance, you may verify whether during some good or bad years, these big groups may attain some actually additional benefits by staying together, but not in all the conditions or across every year. So I wonder if you have any additional information about that.
That's an excellent question, Luis. And we don't yet, because we haven't conducted continuous long-term studies on those sites. So it's something we've talked about doing, and it would be best done in the natural populations where the voles can move around freely, if they need to range further to have access to good vegetation or something, if there's a drought or something like that. Lowell Getz would have those data, but he wouldn't have any data on reproductive success, unfortunately. He would just know who he caught and couldn't assign them to females. So the graph you saw was on a per female basis. It wasn't a group basis. So we've estimated with some of our modeling that there's more offspring born and survive in a group if there's multiple individuals there sometimes. But I think if I remember correctly, that was due to survival of offspring long term. So that's not what I was showing here in this graph. It does seem like there are some benefits to being in a somewhat bigger group. But your point is well taken that maybe there are at some times and not at others. Unfortunately, I don't know. I'm going to have to keep working for another 30 years to answer all these questions.
Yeah, for sure.
Yeah. Thank you for that one, though.
Great. So I'm going to try Marin again and see if it works. She's having some audio and video issues on her end, I think. So I'm still not hearing you, Marin. So I'll get one more question, and maybe you could try and reconnect. But I have her question below. But if Marin, you want to try to reconnect, I'll come back to you after the next question. So I'm going to jump ahead and go to Madison. And that's just a reminder, please, we're trying to build a community. So please identify yourselves and where you're coming from when you ask your question. I always forget to do that myself. So go ahead, Madison.
Hi, I'm Madison Roberts. I'm an undergrad at UTC Chattanooga in Tennessee, United States. So I was wondering, in your enclosures in the Ecology Research Center, if predation was an issue or maybe a benefit to your study, like did that have anything to do with their survival rates?
Good question, Madison. So the voles are in enclosures that are sunk a few feet underground to keep them from digging out mainly. Although they don't read the literature very well, and every once in a while a few dig out. But so we can't, and we have an electric wire we put around mainly to keep raccoons and weasels and some of these out because they'll just eat everybody in the population if they can get them. But snakes could get in. We don't see them that often, but we do have raptor predation quite frequently. It depends on the year. One year there were a lot of rains early in the spring, and so the voles built more surface nests than usual. And we found the radio collars from the voles up in the trees near owl nests and stuff like that. So there were some very, very happy very, very happy owls and some very unhappy researchers that year. And we want some natural predation because that's the point of having these semi-natural enclosures in part, but we don't want predation to the point that we wouldn't be able to collect any data. You had asked if we think this has something to do with some of the effects. It would be hard to answer because we don't know how much predation occurs, and we could guesstimate based on who we don't recapture later. But the voles could die and be under vegetation somewhere, of some other causes.
Yeah, I wondered if like age or maybe food resources would cause a vole to not survive, or if it was predation.
Definitely could be food resources. One year, way before Lauren's time, I had a student doing something with population density in the enclosures, and in one of these enclosures that was at very high density, the voles bred really well, and they ate like all the vegetation pretty much. And so they didn't have hardly any cover at all. And then they died in masses. We didn't find a lot of the carcasses because I think the owls and things picked them off too in that period of time. But we know that a major lack of food or cover can have an effect.
Thank you.
Mm-hmm.
Okay, so I'm going to try Maren one more time. If not, I can read her question. Maren, can you try once more? Okay, so I don't think it's working on her end, so I'm going to just read her question. I think it's a comment, and maybe Nancy, you could comment on this. She writes out, I would like to go back to the common garden experiment. The sample size was understandably low, but the trends were visible. The trends that were visible were in the direction you would expect if there was a genetic effect. So more repeats using both types of ecological conditions, clumped and even, would be great. And it's still amazing to have managed even with just 7 repeats or replicates. So I guess she's looking for a comment based on that. Do you have anything?
Yeah. In the literature that we read, 7 replicates is a lot. Although I understand from a statistical perspective, we're looking at voles within enclosures. So 7 is the sample size. And I agree that it would be good to have both types of vegetation out there, and maybe somebody will want to do something like that. We have at the moment 16 enclosures, so we have a limit unless we build some more on the number of enclosures that exist here. And we'd have to change the schedules up a little bit to be able to handle more enclosures than that, because logistically that would get to be an issue in the heat of summer, getting the voles out of the traps fast enough. But we're starting to switch over to pit tags, and we're going to try, hopefully try proximity collars that talk to each other this summer. So if we didn't have to trap as much, that would be a possibility. Did you want to mention anything, Lauren, about who was heavily involved in building the enclosures?
I don't recommend it.
So one postdocs, Kirkland and Lauren, were I was going to say the major architects, but I think the word I think was major workmen. We were all out there some, but the 2 of them were amazing. And we're talking like clay, solid clay.
Yeah, it was not fun.
Yeah.
um,
The other thing would be to see, I mean, if we wanted to do something like that and build another set or two, if we could if there's vacant fields at the Ecology Research Center, which we'd be able to do that because they're kind of full with studies.
Okay, so I'm going to move on to the next question. This comes from Zulema, please.
Yes, hi. Nancy, great talk. And I'm Zulema Tang Martinez from the University of Missouri in St. Louis, in the US. I'm retired. And Nancy, what I have actually could be relevant to the question Ricky asked about the role of genetics, but this is your show. And I don't want to like crash your show. I could always talk to Ricky afterwards, but if you think it's okay, I can go ahead and say a little bit about a study that I did that looked at genetics.
Sure, that would be fine. And I didn't
Okay.
talk about anything we've done with genetics here today, just there's not time.
Yeah, and I mean, this isn't, this is just looking at relatedness really, rather than any molecular genetic studies, so relatedness. So I did a study that I never got around to publishing. It's on the back burner to write up now that I'm retired. But basically what I had was a large behavior observation room inside. And the study consisted of two types of populations. One population, I had 3 males and 3 females, all unrelated to one another. So that was my genetically unrelated populations. And the other type of population, I had 3 males that were all brothers and 3 females that were all sisters, but males and females were unrelated to one another. I put them in the behavior observation room just loose so they could run around. I had enough nest boxes for each individual and feeding stations for each individual. And then I did a number of observations on them for 3 weeks. And basically what was fascinating is that the population that was, and there were 10 replicates of each. The populations made up of completely unrelated individuals. There were high levels of aggression. Reproductive success was lower. And in the and you never found them grouped together. The most you found was a pair in the nest boxes. So nest boxes got used, but only by pairs. In the related groups, it was just the opposite. Virtually every time that I looked, you would find 5 to 6 individuals in each nest box. So they were really crowded together. And the cases where I only found 5, usually it's because one ran out as I opened the nest box to look in. And they had higher reproductive success and very low levels of aggression. So I think that that's, obviously a brief summary, but I think that this might be relevant to the question that Ricky was asking at the beginning.
Thanks,
But again,
Elena.
yeah.
That jogs my memory. There was a student of Mike Gaines out in Kansas, I think did a study for her PhD. I can't remember the details because it's been a long time, where she varied the relatedness of, I think, the females, or maybe she just looked at the relatedness of the females in the population and found that there was some effect, but I can't remember what.
Yeah, that was Wendy Sarah. And
Right.
The 2 studies are very similar. And I was doing a sabbatical with Mike at the time. And I can't remember the exact results. And I think you're right. I think she only looked at females, and it was an outside enclosure. So she wasn't able to monitor actual interactions. It was just looking at some demographic aspect of it. But I think certainly the results were consistent. In my case, because it was a behavior observation room inside, I was able to get more details on the interactions among the voles and more details on how, they were living and so on. And it was like really dichotomous. I mean, I never found more than 2 voles together, male and female, in the unrelated group. And virtually every time that I monitored the groups, I found virtually all of the individuals in the related groups all jammed together in one little tiny nest box. So I don't know, Ricky, if that has relevance to what you were asking, but I suspect so, maybe.
Yeah, very, very much. Am I allowed to ask a follow-up question to that? Is that okay? Or should we talk about this later?
Sure, go ahead.
Okay, really briefly, do you think that thank you so much for sharing that. That is extremely relevant to what I was thinking. Do you think they still formed pairs? Or were they a cohesive 6-vole unit?
Yeah, I can't say, I mean, I sort of assumed that they did, but obviously I can't say from my data, and we didn't do any genetic analyses, so I can't really answer that question.
Thank you. Thank you very much.
Sure.
That's okay.
We should have
And
some
they-
data maybe by this winter too, Ricky. We've got an undergrad here that's looking at vegetation near nests and randomly and at random places in our enclosures. She's going to look at group sizes, group composition. So she's going to look at relatedness, reproductive success, et cetera. But it's going to be a while until that all gets done.
And again, Nancy, super talk.
Thank you.
Thanks.
Fun to find out what we did. I didn't realize after a while you start to forget some of your own work or some of the details of your own work.
All right, so I'm going to move on to the next question, and that's from Becky Rosengass.
Can you hear me?
Yes, you can. Thank you.
Hi, how are you? Nancy, thanks so much for your interesting talk. I'm Becky Rosengaus. I'm working with termites, and I work at Northeastern University here in Eastern United States. And Nancy, this question is more of a methodological question. I'm very intrigued about the fluorescent powders that you're using for your labeling of these little guys. And I'm wondering, I've been trying to actually label termite eggs, and I'm wondering whether these powders, I mean, you gave me an idea as you were talking about, whether you can use these powders to label insects or perhaps insect eggs, and whether these things are toxic or not. What if they ingest it? I am sure the termites will be feeding on this powder, and I'm wondering whether I'm going to be killing them, or is it okay for them to eat it?
Well, I could answer part of your question. I know I've lent some powder to people here at Miami University, and they have dusted insects. And then they'll lay down the trails as well that they could follow. I don't know about the eggs. I can, if you're at Northeastern.
Yeah, I
What
can
department?
shoot you an email maybe later.
Yeah, because we haven't had to buy powder for a long time. I can see if the same company still sells it. If not, maybe I could send you some little baggies.
That would be awesome. Thank you. Thank you. Do they come in different colors, you said, right?
We have different colors.
Uh-huh.
We didn't buy them all because some look too much alike at night, and some you can't see in the grass as well. Like the yellow greens don't show up very well. But I could look at the cans and see if it says anything about toxicity. Now, the voles also groom each other.
Mm-hmm.
And I mean, we've really coated them. So,
Wow.
you know, they're much larger than the insects and things.
Well, great. If you don't mind me bothering you later through email.
Not at all. Not at
Thank
all.
you.
Happy to help.
Thank you so
It might
much.
also come as a paint. Because I think
Yeah.
this company also made something that wasn't in a powder form.
Okay, we'll talk about it. Thank you so much. It's a good idea. You lightened my head today, like, oh, little light bulb went on. Thank you.
Right. Good.
Okay, so we have the next question comes from Caleb.
Hey, Nancy, great talk. My name is Caleb Vogt. I'm a PhD student at Cornell University in the lab of Dr. Michael Sheehan and Melissa Warden. So I have two questions. So my first question is about male and female differences in actual spatial or patterns of space use in the enclosures over the field season. So I know you guys are using like a trapping system and then the RFID system. And my question is, to what extent have you used the RFID system in the semi-natural enclosures to get a sense of like, to what extent are males and females being detected at unique zone locations or RFID locations in the enclosures? So for instance, are males mostly staying in one particular area of the enclosure, or are they moving all around, or are females moving all over the place? I was wondering, I think you had some data showing that the males, there's some statistical difference in the amount of, or the distance that they're traveling, but I don't think you showed any data on like where they are spending their time in the enclosures. And is that stable over time, or does it fluctuate? So that's my first question.
Okay, good. Yeah, let me answer one at a time before I forget what you ask if you ask too many. So, Lowell gets, and we have, and I'm pretty sure that the Ophir lab as well, describe male voles in particular, sometimes females as well, but particularly males, as being either residents of a nest, so they have a smaller home range, centered, or maybe not centered, but the nest site is within the range. And then there's other voles that, if we're using the multiple capture live trapping or we track them, have larger home ranges and they overlap multiple nests, and we call them wanderers. So it's these alternative reproductive tactics that have been described in a number of species. And we've looked at those as well. So if a male's a resident, he tends to stay, as far as we could tell, in a smaller home range centered around his nest, and the wanderers overlap larger portions of the enclosures in multiple nests. And I think that's what you were asking.
Yeah, great, thank you. And then the follow-up to that would be, do you guys have any data on what happens when a male who happens to be sort of stationary or has a single nest, what happens when they either gain or lose a partner? Is there any data on like their spatial movement patterns after an event like that? Either again, the gain, so presumably actually gaining a partner probably leads to the formation of that stickiness to a territory, or maybe I'd be curious to hear what your thoughts on that, on what comes first, but especially in the event of a loss. Either, I didn't realize that your field enclosures had active aerial predation, so I don't know if you guys have data on like female partner loss or male partner loss, but how does that affect the spatial patterns of movement for either sex?
Want to do some data analysis with me? So we've got the data to answer your question in your spare time, right?
Right.
So we've got the data that we could answer that question, but I don't know. We do definitely see losses of males or females. Sometimes they move, but more often they get predated. And Lowell Getz had said, and then he and Sue Carter did some lab work showing that the males, at least right away, did not form new pairs. But I think that's not true over a longer period of time. But we definitely we don't know the answer right now, but we have the data that could address this.
Thank you so much. I appreciate it.
Okay, and there's a student who's working in the enclosures now, and part of what she's going to do, and she's waiting to start. First, she's locating the nests, and then she's going to do removals in some of these enclosures. So either remove a male or a female and look at what happens, or remove both the male and female and see what happens to with the voles close by, or even not so close by. So that would really be able to answer that question.
I might slip one more question in there because I just remembered it. Do you provide any food resources or water resources in your semi-natural enclosures, or is it completely just whatever material is there? And you may have already said this. I might have forgotten.
No, I didn't say. We once upon a time, yeah, we did do some supplemental feeding manipulations, but in general, we've planted the enclosures so there's sufficient amount of dicots there for the voles to survive, and they get their water either through metabolic water or from rain on the plants and things. So we don't add either.
Thank you.
Sure.
So I think I actually have a question that follows up on the theme that Caleb was bringing up to you, Nancy. And I apologize if you've answered this. I'm juggling like 4 different things, so I'm not totally listening to everything that's being said. So I think in terms of like Luis and I have shown with the degus that stability is really important in the fitness to both males and females. And now I'm talking about group stability. And you mentioned changes, removals of males and females. Have you looked at how stability influences the reproductive success of both males and females? Do you have the data to do that?
We do. Let's write a paper. So yeah, so I'm well aware of what the two of you have done, and we've never looked at those data. So we have groups that remain stable for a period of time, and then some in which there's losses or additions or movements. And we've got the parentage data, but I don't know the answer. Ooh.
And the other question I have is, have you thought about, and again, I apologize if I missed this in the talk. The other thing we've seen is that males are problematic to females in degus. In the larger groups with more males, females seem to suffer costs.
Yeah.
Do you have the kind of information that would allow you to look at sort of male group composition and how that would impact the females?
Yep, because we know who, based on the nest trapping and either telemetry or whatever else we've used, we know who's living with whom.
Yeah.
So we could get a pretty good idea of group composition. And then we've got data on reproductive success and things.
And you have maternity and paternity, right?
Yes.
Okay.
Both. And for the enclosures, we're able to assign up to 100% sometimes of offspring to the parents.
Because it seems to me like the nice thing about the enclosures is you can look at some of those relationships that we can't look at in the field, just our natural population, because you can look at how change in density influence the stability of groups and how that then plays into fitness. That'd be really pretty cool, I think.
We could do that.
Yeah. Need a postdoc.
Or a PhD student. Since I'm going to work for another 30 years, I guess I've got time. Or both. Time to repopulate the lab.
If you have other questions, I don't have any in the queue right now. I'll wait just a moment in case anyone has a follow-up or that question they didn't, were a little bit afraid to ask, or if not, I'll end the live stream and just give people just a moment or two, and then if not, I guess I'll stop the live stream. We can keep the conversation going. And before I do, just let's thank Nancy again. Thank you, Nancy. That was really awesome to hear about all that work. Especially fun for me to hear about what you guys have been doing since I left scenic Ohio. And so at this time, I'm going to stop the live stream